Hello students, good morning all of you. We already discussed about the multiple alleles. Okay, the multiple alleles means a gene, it has generally more than two alleles. Generally the gene has the two alleles, but when there are the more than two alleles, then it is known as the multiple genes. We already discussed about the blood system, it is ABO blood system in the human being. Now today we are going to discuss about the eye color in Drosophila. Another example, that is eye color in Drosophila. Okay, it is observed that the gene W, it is for the eye color, it is the X-linked gene. It is X-linked gene. So the red color of the eye, it is the wild type, or this wild is also known as the dominant. Okay, it is also known as the dominant. And another, that is the white, and it is the white is the mutant, or it is the recessive. Okay, so these are the two normal dominant and recessive types of this alleles. The red is dominant and white is the recessive. So, or the red is the wild and the white is the mutant one. Okay, but but in F2 it is observed that there are the dozens of the colors they are observed in the Drosophila population. Okay, dozens of the colors they are observed in the Drosophila population. Which are the dozens of the colors? So first of all, the red color, okay, it is denoted by the W. Okay, then after that there may be the different colors like coral, they denoted as the CO. Then the blood, it is denoted as the BL. Then the eosine, it is denoted as E. Then cherry, okay, the cherry, it is denoted as CH. Then apricot, it is denoted by A. Then honey denoted by H. Then buff, so it is by the BF. Okay. Then pinch, it has denoted by P. Then pearl, that is P, and ivory, that is I. So these are, and the last one, that is the white. So these are the 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So these are the dozens of the colors they are they are present in the Drosophila population. If we study the hierarchy, so this hierarchy is denoted as the W plus, which is the dominant, therefore it is the plus, or it is the wild type. This wild type is dominant over the coral. Coral, these are the different colors, these are the different shades, okay, of the red color, and in between the red color and white color. Okay, so this coral color is dominant over the blood color. This blood color is dominant over the eosine. This eosine color is dominant over the cherry. This cherry is dominant over the apricot. Apricot is dominant over the honey. Honey is dominant over buff. This buff is dominant over peach. then the finch is dominant over the pearl and the pearl is dominant over the ivory. Okay, and all these shades they are dominant over the white. Okay, so these are the 12 different shades of the eye color in Drosophila, and they shows the dominance hierarchy. That is, the red is the dominant one, and these are the different color shades of the eye color in Drosophila. Here we will see one figure. So these are it is the wild color. It is the wild or the red color. Okay, and these are it is the white color. It is white color. It is red color, wild red color, and these are the intermediate shades between these. Okay, so so it is the example of the Drosophila eye color. So the scientist Lewis, he stated that the sum of the alleles in the multiple series, it may be the pseudo allele. It may be the pseudo allele. That means it is not the perfect allele, but it is the intermediate development of intermediate color or intermediate type. Okay. So according to the Lewis, the sum of the pseudo alleles has been present in the population. Okay. I hope you got the point. You all these color shades you may check it on the internet. Okay, color shades, what types of the color shades. Okay, now we will discuss about the second example. That example is the cell incompatibility in Nicotiana, or it is also known as tobacco. Okay, so in self incompatibility means what? Sometimes it is observed that each plant it produces the pollen grains as well as the ovules. Okay, so the anthers and the stigma, gynoecium, they are present in the same chromosome. So when pollen they falls on the stigma, okay, so this pollen germinate, it produces the pollen tube, and the male gametes reaches up to the stigma and the fertilization takes place. But it is observed that in some of the plants, or particularly in the Nicotiana, we are discussing about the Nicotiana. So in Nicotiana, it is observed that when the pollens of the same flower, when they fallen on the ovules of the same flower, they do not fertilize. Okay, the same flower, the same flower, same flower, pollen grain and ovules, they do not fertilize. They do not fertilize. So are they sterile? No. When these pollens they fall on the another ovules, okay, the ovules of another flower, they show the fertilization. They show the fertilization. Or the another plant's pollen, when they show fertilization with another flower ovule. Okay, so that means these pollens and these ovules of the same plants, they do not fertilize. That means they are self incompatible. They are self incompatible. Incompatible, that means they are not suitable to each other for the fertilization. So this self incompatibility is developed in the plant. It is to avoid the self pollination. To avoid the self pollination and to promote cross pollination. To promote cross pollination. Okay. So it is observed that in the scientist quadrature, it is observed according to the scientist quadrature in 1764. The scientist quadrature in 1764 he described the same self-incompatibility. He described the self-incompatibility in Nicotiana, in tobacco or in Nicotiana. Okay, so this phenomenon is also observed in many of the monocot and dicot plant also. In many of the monocot and dicot plant also this self incompatibility is observed. Then the scientist East and Mangeldorf, East and Mangelsdorf, Mangelsdorf in 1925 they discovered the number of alleles for this S1, S2, S3, S4, S4, S5 and so on up to Sn. Okay, so the number of the alleles which are present in the plants or it is or these alleles they are present and these alleles they are labeled as S1, S2, S3, S4 and so on. Okay, so the Nicotiana plant it could have the sum of the alleles or a single plant it has the only two alleles. A single plant it has the only two alleles whatever may be the combination. Suppose there may be the S2, S3, suppose it may be the S1, S2, suppose it may be the S1, S4, whatever may be the combination. The single organism it has only two alleles. Okay. In this way, there are the number of alleles which are present and accordingly that it shows its incompatibility. Okay. Suppose there is a plant which have the S1 and S2 allele. The pollen grains of this it has the S1 and S2 alleles, okay, of the organism. The organism, suppose it is the male, it has the S1 and S2 alleles. So it produces the two types of the pollens S1 and S2. Okay. So similarly, the female, it also have the S1 and S2. So it also produces the S1 and S2 gametes. Okay. So it is observed that both have the similar alleles and therefore the similar allelic gametes they are produced by the pollen as well as the these are the ovules. Okay. These are the ovules. So in that case there is no fertility, no fertile. So no seed will produce. No seed will produce in such organisms. Okay, okay. So there are, we will draw here one table. Okay. So here the genotype of parent, here the functional column and these are the resulted progeny. Okay, and this one serial number. So first of all When S1, S2, it is crossed with the self S1, S2. Okay. So there is no functional colon. No functional colon. Okay. Or we can say that none. None. So zero seeds will produce. Okay. Zero seeds will produce because S1, S2, S1, S2. The second case where the S1, S2 cross with the organism which having S1, S3. Okay. So it is the female, it is the male. Okay. So here the functional colon is only the S3 because SS is the common. The ovule S2 and S3. These are the two things. Okay. So it produce the S1, S3 organism progeny and second S2, S3. Okay. So this S1 is formed with S3 and this S2 is formed with S3. So these two progenies will produce. Then the third case S1 is crossed S1 S2 organism is crossed with S2 S3. Okay. Here the 2 is the common and therefore the functional colon here only S3. Okay, S3. So it produces the organism S1 and S3. Okay, S1 and S3. S2 it cannot be taken because S1 and S2 they are there. Okay, so S1, S3 and the S2, S3. Okay, S1, S3 and S2, S3. Okay, so these organisms will produce or these proteins will produce. Then the fourth case, suppose there the organism S1, S3 alleles, okay, and they crossed with the S1, S2. Okay, they crossed with S1, S2. So only the 2 is functional colon there because it is the male and it is the female and therefore the functional colon S is the common in both of them and therefore only the 2 is the functional colon. So S2. Okay. Then the organism will produce that is the S1, S2. Okay. S1 and S2 and then second S2 and S3. Okay, or you can write that S3, S2. So it is the case. Then the fifth one that is the S1 and S3 when it is crossed with the S2, S3. Okay, so 3 is the common here. So only the 2 is the functional. So only S2 is the functional. So the S1 is produced with the S2 and this S2 is also crossed with S1. This S1 is crossed with this S3. Okay, and then S1 is also crossed with this S2. The 3 is common, therefore do not produce the progeny. So S1, okay, S1 is the function. So S1 and S3, okay, and S1 and S2, okay. Then the sixth case, S2 and S3. This organism, female, is crossed with S1, S2. Then here only the S1 is functional. So it produces the progeny S1, S3. Okay, the two is the common. So S1 S3, okay, and then S1, S2. This S1 is from one time this and one time with this. Okay, S1, S3 and S1, S2. Then the second case, S2 and S3 is crossed with S1 and S3. Okay, so S1 is the only functional, so it produces S1, S2 and S1, S3. Okay, this only this functional, so one time this and one time this. Okay, S1 with S2, S1 with this S3. So generally these combinations will produce. So it produces only two progenies. Okay. So we will draw here one figure. Suppose it is the gynoecium. Inside, okay. So on the stigma, there are the pollens situated on it. Okay. So and the ovules we will denote it by the red color. So it has the S1 and it also has the S2. Similarly here also the S1, S1, S2, S2. Okay. So it will not fertilize. Okay, it will not fertilize. Why it will not fertilize? Because both they have the pollen grains also have S1, S2 and the ovules also have S1, S2. Okay, so no progeny, no progeny will produce. Okay, then the second figure. Okay, so the pollen grains, okay, S2 and S3. So S2 and ES3. Okay, and the ovules, S1 and S2. This is S1 and ES2. Okay, so the functional pollen S2 is common. So functional pollen is only the ES3. Okay, so this S1 it may produce with this S1. This S2 it will produce with this S1 and this S3 may produce with this S2. or it may be with the S1. So it will produce S1 and S3 as well as ES2 and ES3. Okay, this ES1 and ES2, so ES1, ES2 and this ES2 with this ES3. Okay, so these two progenies will produce only. So the first, okay, so here in this case the pollen grains they are ES3 and ES4. 1, 2, 3 and 4. So they are ES3 and ES4. Okay. And the ovules are ES1, ES2, ES1, ES2. So these are the ES1, ES2, ES1, ES2. So these are the ovules. Okay. So this ES1 is produced will pollen from this. Then this ES3 is also get produced from this ES2 and this ES3 also produced. Then the ES4 and ES1 is also produced. Similarly, this ES4 is also produced. So it will produce the ES1, ES3, then ES2, ES3, then ES1, ES4 and then ES2, ES3. So, ES4. Okay. So these four progenies are produced. Four progenies will produce. Here only two. Here no, zero. So such cases, these are the semi-compatible. These are semi semi compatible. These are the fully compatible. Fully compatible and they are self incompatible. They are self incompatible. Okay, such the different types of conditions or different types of the alleles they show the fertilization with the different allele organism. So it is the example of self incompatibility in the Nicotiana or tobacco. Okay, so the multiple allelism means the single gene it has more than two alleles, so that is known as a multiple allelism. We have discussed three examples about the multiple allelism. One is the ABO blood group system in the human being. The second that is the eye color in Drosophila and the third, that is the self-incompatibility in the tobacco or Nicotiana. I hope you got the points. Thank you very much.